अपने लेक्चर को थोड़ा सा मॉडिफाई किया है, ठीक है. इसको PCOs पे वी विल बी टॉकिंग अबाउट PCOs, पॉलीसिस्टिक ओवेरियन सिंड्रोम और पॉलीसिस्टिक ओवरी सिंड्रोम इट इज़ ऑल्सो नोन एज. व्हाट इज़ दिस, अ लिटिल बैकग्राउंड, और हम इसको आइडेंटिफाई कैसे करते हैं और इवेंचुअली हम इसमें हैंडलिंग कैसे करते हैं, समेयर कैसे बनाते हैं. वी विल बी टॉकिंग अबाउट, वी विल बी कंसिडरिंग अबाउट दिस थ्रू आउट आवर डिस्कशन और टॉक. PCOS and its diagnosis. What PCOS is, polycystic ovary syndrome. Basically, this is an endocrine disorders. This happens due to the disturbance in endocrine system, and it affects the female of reproductive ages around 5 to 10%. It is basically characterized by presence of cyst fluid-filled cavities in the ovaries, and this leads to the infertility in female, and this is a very grave issue, and no particular treatment has been discovered so far. General symptoms: irregularity in cyclicity, hyperandrogenism, disturbance in androgens level, male reproductive hormone testosterone level increases in female. Ovulatory dysfunction is also a very common symptom. Insulin resistance and psychological and emotional disturbance is also a very common symptom of PCOS. Then underlying factor, this is basically a pleiotropic trait. It is controlled by many factors. Genetic factors are there, hormonal disturbance, as I have previously told you, increase and decrease in testosterone level, and exposure to various endocrine disruptors, chemicals that are present in the environment, as well as obesity, as well as sedentary lifestyle. These are all the factors which cause, which are major underlying factor behind the PCOS. Next, potential complications. This leads to infertility, type 2 diabetes, sleep disorders, gestational diabetes, ovarian cyst, hirsutism, and acne. These are all potential complications due to the PCOS. Next, experimental study on PCOS. Basically, experimental studies are conducted on model organism. Although they have estrous cycle, rats have estrous cycle, but it is very easy to handle rats, easy to handle. They have very shorter reproductive cycle. They have the same sort of mechanism, hypothalamic pituitary gonadal axis, HPG axis. So this is easy, and it gives up to some extent reliable results. So that's why we choose rats as an experimental model. Next, what we do actually, we divide animal into the various group. One is considered as control group, that is not showing any symptom of PCOS. Second one, the PCOS group, that have been introduced PCOS via various method or various strategies. There are two ways to induce PCOS. One, by giving them fat-rich diet. Secondary, by giving them letrozole. So we often use letrozole. Letrozole is aromatase inhibitor. This results in increase in testosterone level, and this eventually cause the PCOS, and females become infertile. Laboratory environment, we generally consider temperature, that is generally kept at 22 to 25 degrees Celsius. 12-hour day and dark and light cycle is also maintained. Humidity is maintained between 55 to 60 degrees Celsius, and free access to tap water and feed char is usually given. Next, handling of rat during this process. Usually for taking smear, which we are going to discuss in subsequent slides, how we basically capture the rats, how we deal with them, how we handle with them. Scroofing method is generally used for entire purpose. We just capture the rat from the upper foreskin, then skin is tightened, then we catch it very tightly, and then we use our other hand to stretch its hind limbs. Then its vagina is exposed to take the smear. Steps are shown here. First, rat are caught from their foreskin on upside on their neck. Then they are stretched. and then their vagina is somewhat exposed to take the smear. Vaginal smear collection. Basically, we are introducing PCOs because that is for various purpose, for the treatment purpose, for to know about the genetic reason, to know about the cause. That depends on the experiment, what we actually want to do. So how we know whether PCOS has been generated or not? How we come to know? हमें पता कैसे चलता है? देखिए, हम तब तक उसके molecular analysis के लिए नहीं जा सकते जब तक हमें पता नहीं चलता कि at the first step whether PCOS has been generated or introduced or induced or not. Vaginal smear taking is one of the easiest and reliable method to know either it has happened or not. What we do, we actually capture the rat by the method I have told you in a previous slides. Then there are two ways. One, either we can take cotton swab, or we can take dropper, or we can take micropipette. So we take saline into it and we inject that saline into the vagina of the rat, and then we suck it back. Now it collects fluid as well as cells from the vagina. Now those fluid has been transferred into your pipette or has been attached with your swab. Now you transfer it on the slide and you stain it with Mayer's hematoxylin. And after staining, you will be able to gauge or measure or judge whether PCOS has been generated or not. This is diagrammatic representation. The person is having female rats in his hand. Vagina has been exposed. Now the saline has been introduced or has been poured inside the vagina, and now it is asphyxiation is taking place. It is sucking back. Now it has been transferred onto the slide, and subsequently it has been stained. तो ये वो जो सारी बातें मैंने आपको पीछे बताई, मैंने उसको डायग्रामेटिक उसके स्टेप बताए कि हम हैंडल कैसे कर रहे हैं. फर्स्ट वही माइक्रोपाइपेट को लेते हुए आपने क्या किया, इंजेक्ट किया सलाइन के अंदर आपने जो है वो सेल्स कलेक्ट कर लिए, ठीक है. अब नाउ यू हैव प्लेस्ड इट ऑन टू द ग्लास स्लाइड. नाउ Mayer's hematoxylin is used to stain. Now there are 4 important stages of female reproductive system in rat. One is proestrus, other is metaestrus, other is estrus, then metaestrus, and at the last is diestrus. How we characterized whether this stage which we are observing is proestrus, metaestrus, what is this? Actually, in smear there are different types of cells. In proestrus, the slide is enriched with nucleated cells and round cells. Proestrus stage is a stage where the level of estrogen is increasing, not up to its peak, is increasing. So at this stage, the nucleated cells are at their peak. Most of the cell in smear you will find the nucleated cells. They are round and those are very clear cells. Then comes the estrus stage, as it is indicated by the name. At this stage, estrogen level is highest rather. At this stage, there are very few nucleated cells, and this is enriched with cornified epithelial cells. More and more cornified epithelial cells would be there. You will be able to observe those cornified cells. Proestrus stage lasts for 14 hours, while estrus stage it lasts for 1 to 2 days, right? Then comes the metaestrus stage. This lasts for 6 to 8 hours, and this is enriched by 3 types of cells, entailing nucleated cells, cornified cells, as well as leukocytes. When in smear we find these 3 cells in equal proportion, we will be able to infer that the animal or the female animal is in metaestrus stage. Then diestrus stage, this is enriched by nucleated cells as well as the leukocyte. Cornified cells are very few in this stage. Microscopic examination, we make the slide, we spread the smear, then we stain it, then we observe under the microscope under various magnification, and we observe various stages. This is diagrammatic representation, proestrous stage, enriched with nucleated cells. Next, the estrous stage, cornified cells, metaestrous stage, 3 type of cells, and diestrous, predominated by nucleated as well as the leukocyte. Now, what happens in normal rats and what happens in those rats which are undergoing through the PCOS? This is a very good diagrammatic presentation showing the normal female rats as well as the rats undergoing through the PCOS. The red line indicating the normal female rats and the blue line is indicating the female rats. Those have been induced with PCOS. As I have told you in the first stage, proestrus, that in female rats that lasts for 14 hours. Next stage lasts for less time, that 1 to 2 days. Next stage, metaestrus, lasts for 6 hours, and the last stage lasts for the 3 days. So in the red line showing there is a proper downward curve, there is a proper specific time for which animal in a specific stage. While if you consider the PCOS, you will be able, those time duration animal should be in that stage that is not being completed here. The animal is in that stage for a very short span of time. Even in some time, one of the particular stage is missed. So that's why there's not proper reproductive cycle, and that leads to the infertility. Next, that is further now from just smear we are able to identify, we are able to detect कि जो animals हैं वो PCOS has been induced. अब नेक्स्ट हमने क्या करना है, उसकी कोई जेनेटिक स्टडी करनी है या कोई फार्माकोलॉजिकल थेरेपी यूज करनी है, व्हाटसोएवर वी हैव टू डू, देन वी डाइसेक्ट द एनिमल्स or the basic analysis which do is the there are various molecular analysis we perform, but here I have only showed the histopathological analysis के वहाँ पे हमने उसको formalin में we preserved it and observed it under various magnification. These are the diagram picture photomicrographs, the control group showing the normal histopathology histology. Various stages are very clearly shown. Graphian follicle, उसके बाद primary follicle, secondary follicle, tertiary follicle, corpus luteum, means everything is in order. Everything is being presented into a very, very normal and order way. While the B diagram, this belongs to our article. In the second, it is very evident. You will see there is fluid-filled cavity or cyst present there. Those are very clear. So our first examination, smear analysis, has been further endorsed by this diagram. This shows that PCOS has been properly introduced or induced into the animal. Next two photographs are from the other group which were treated with various flavonoids, and we are able to observe that PCOS has been controlled, or PCOS has been taken over up to the very effective extent. So this is how we do work on PCOS. We introduce PCOS. We perform various analysis, genetic analysis, molecular analysis. Even we can go for messenger RNA silencing to check the effect of hormone. Then hormone therapy comes. Then there different sort of food treatments come. Then the obesity related issues are also considered and related with this research work. Okay. Thank you so much. Any question? जी जी basically that affects the hormonal axis. हार्मोनल जो है वो डिस्टर्बेंस आती है। जैसे ही हार्मोनल डिस्टर्बेंस आती है तो वो रिप्रोडक्टिव साइक्लिसिटी जो है वो जो है मेटाबॉलिक डिसऑर्डर है तो उससे रिप्रोडक्टिव साइक्लिसिटी डिस्टर्ब हो जाती है। जी बिल्कुल इस सही कह रही हैं जी। पीसीओ हम सिर्फ पीसीओस को ही कंसीडर करते हैं, जो आप कह रही हैं उसको हम कंसीडर नहीं करते।
ये नई टर्म आई है बेटे, ये नई टर्म आई है पॉलीसिस्टिक ओवेरियन सिंड्रोम है। पॉलीसिस्टिक ओवेरियन डिजीज एंड पॉलीफोलिकुलर डिजीज इज द सेम। मतलब जब हम सिंड्रोम की बात करेंगे हम ये कहेंगे कि मल्टीपल ऑर्गन्स इन्वॉल्व हैं, उनके सिम्टम्स हो रहे हैं तो सिंड्रोम कहेंगे। अगर वो क्रॉनिक हो जाएगी तो उसको डिजीज कहेंगे। और अगर हम ये न्यूली स्टडी करेंगे ना, रिसर्च जो है तो इसमें पता चला कि ये जो हम सिस्ट कह रहे हैं ना, ये एक्चुअली में इमेच्योर फॉलिकल्स हैं। उसका नया नेम जो है वो पॉलीफोलिकुलर डिजीज है। ठीक है, तो जिस स्टेज पे आप इसको स्टडी करोगे वैसे ही इसका वो नेम बेसिकली इसके सिम्टम्स और
जी अभी तो इसमें वही मेटफॉर्मिन और ग्लूकोफेज वगैरह ही हम इसको देते हैं। इसका कोई एज सच जो है वो ट्रीटमेंट कोई डिस्कवर्ड नहीं है। ठीक है, अगर वो ज़ाहिर है इंसुलिन रेजिस्टेंस है तो उसको ग्लूकोफेज वगैरह देते हैं या मेटफॉर्मिन देते हैं, उससे उसको मैनेज करने की कोशिश की जाती है। इसकी प्रॉपर जी जी वो मल्टीपल रीजंस बताई हैं, सिर्फ ओबेसिटी इज़ नॉट वन ऑफ द रीज़न। बाद में भी ओबेसिटी जो भी ओबेसिटी हो सकती है, जेनेटिक फैक्टर हो रही है डाइट की वजह से। बिल्कुल डाइटरी चेंजेस, सेडेंटरी लाइफस्टाइल। नहीं, ये इसका तो अभी साइंटिफिक कोई एविडेंस नहीं है। अभी जैसा मैडम ने भी बताया कि अभी तक हम मेटफॉर्मिन
बेटे आप किसको कह रहे हैं, फर्स्ट 14 डेज में आपने स्टेजेस को देखना होता है, नेक्स्ट 14 डेज में आपने प्रोजेस्टेरोन को देखना होता है। फर्स्ट 14 डेज में आप सेसमी सीड्स देते हैं और फ्लैक्स सीड्स देते हैं और नेक्स्ट में आप देते हैं। तो जो ओवेरियन डिसफंक्शन है ना, वो देखना पड़ता है कि उसमें आपके साइकिल मिस हैं या आपको ज़्यादा ओवर फ्लो के साथ आ रहे हैं। पॉलीसिस्टिक ओवेरियन में दोनों प्रेजेंटेशन हो सकती हैं, ओवरवेट भी हो सकते हो आप, अंडरवेट भी हो सकते हैं। तो सीड साइकिल जो है वो पेशेंट के सिम्टम्स को देख के रिकमेंड होता है, उसकी हिस्ट्री को देख के। हर एक के लिए नहीं होता कि आप नेट से देखें और आप खुद फॉलो करें। नहीं, नो डाउट सीड्स जो है वो हेल्दी होते हैं, बट अगर आपने हार्मोन पर फोकस करके लेना है तो उसकी अमाउंट, टाइमिंग और अपने सिम्टम्स को देख के आप लोगे, वैसे नहीं लोगे।
अदरवाइज तो मजीद डिस्टर्बेंस वो कॉज कर सकते हैं। थैंक यू।
Thank you so much, sir, for your valuable talk. As we gather to explore the art of animal medicine, we are delighted to have Assistant Professor Dr. Shahab Irfan join us as a keynote speaker. Your wisdom and insight will undoubtedly guide us toward a deeper understanding of these magnificent creatures. Please join us on dais. सर माइक है, रिकॉर्डिंग हो रही है, मतलब सही बंद है।
मुझे थोड़ी सी आपकी अटेंशन चाहिए होगी, बैक बेंचर्स प्लीज़ थोड़ा सा. ओके सो अपनी प्रेजेंटेशन में आज मैं आपसे टॉपिक डिस्कस करूँगा. उस टॉपिक इज रिलेटेड विद दी diabetic models. Or diabetes is specifically a problem of islets of Langerhans. Islets of Langerhans, they are present in the pancreas and they are normally present in the mammals. So the title is Islets of Langerhans, rodent versus primate. Differences in cytoarchitecture and subsequent metabolic signatures. The reason for this presentation is that we use mouse or rat models in diabetes. And how relevant these models are with the humans, because diabetes is a human disease. Diabetes naturally does not happen in animals. So how relevant this rodent model is to the humans? Okay, so islets of Langerhans, I will explain briefly what are islets of Langerhans. Islets of Langerhans is actually an organ within an organ. So these are a separate kind of a functional entity inside the pancreas, and the pancreas has an endocrine portion and an exocrine portion, and in that organ these are a independent endocrine entity. Islets of Langerhans, they are responsible for the homeostatic control of the glucose, and what I mean by the homeostatic is that during the feeding and during the fasting. So during the feeding we need more insulin, and during fasting we do not need insulin. So both these physiological conditions, feeding and fasting, are controlled by the islets of Langerhans. Insulin secretory beta cells and glucagon secretory alpha cells, they work in tandem and to control the plasma glucose levels during feeding and fasting. Glucose concentration in our blood acts as a main trigger for the insulin release, and the lack of glucose in our blood acts as a main trigger for the glucagon release from the alpha cells. There are cells in a single islets of Langerhans, and I don't think you will be able to see it fully, but there is a negative correlation between the glucagon levels and the insulin levels. Okay. So if insulin levels are high, it always means that the glucagon levels will be low, and if the glucagon levels are high, it always means that the insulin levels will be low. So this is the normal physiological mechanism of homeostatic glucose control. Now, why islets of Langerhans are kind of unique in their structure? I would use the word royal, so I would call them a royal enterprise, and then I will explain why they are a royal enterprise. Till date, not a single pituitary hormone has been discovered which can modulate islets of Langerhans. So these islets of Langerhans, they are hypothalamic pituitary independent endocrine tissue. Not a single pituitary, anterior pituitary, pituitary or hypothalamic hormone can actively regulate alpha cells or beta cells. So, and the general notion of endocrinology is that all of our endocrine glands are regulated by pituitary hormones. So this is a very unique, independent situation which these isolates of Langerhans they have. Apart from this, these isolates of Langerhans they are strictly endocrine. The mode of cell communication they go through is autocrine, juxtacrine, intracrine, and endocrine. So we have no issue in understanding them as an endocrine organ, but they behave, they act independent of the pituitary. So they have a local regulation. Now, in our experimental modalities, we use rodents because they're easy to handle, they're small in size, we can recruit as many animals as we can, and we can develop multiple types of different physiological models. Same is like this, we also use a lot of murine rodent models in these in experiment regarding the diabetes. And there are two types of diabetes, type 1 diabetes, type 2 diabetes. So we can generate both of these type 1 diabetes models and type 2 diabetic models in either rats or in the mouse, which is fine. And the most common methodology is that we use two chemicals, namely Alaxon and streptozotocin. Both of these chemicals, they are similar in their mode of action, kind of, not exactly, but kind of, and both of these, Alaxon and streptozotocin, they specifically destroy beta cells. They cause beta cell apoptosis, and this makes a normal animal, a normal rat or mouse, into a diabetic rat or mouse, and they will have less concentration of insulin, which results in a very high level of plasma glucose. So this animal is now hyperglycemic, and over time it will become diabetic. This mode of diabetes induction is considered gold standard. Okay. Now, these rodent or marine models, they are, because they're extensively used, so a huge wealth of data has been generated over the last 20-30 years. And there should come a question that how relevant these rodent models are with the humans. Or why we should ask this question, because the last point, the success ratio of our clinical trials. So we discover a new pharmaceutical compound which is very helpful in diabetes. So how, and we test that pharmaceutical compounds in animal models, and it is very successful, and we move on to the human trials. And the success ratio is extremely low. I will show you the most recent data, that is 2019. This last, so this last bar over here, this is the ratio of success from phase 1 to actually approval. So this is around 4.9%. So if 100 chemicals are selected for diabetic treatment, out of these 100, only 4.8 of these chemicals will be approved. So this is a success ratio of just 4%, or failure ratio of 96%. And the main reason for that is that these compounds were actually developed while working on animal models. While working on the animal models. So, what could be the problem? So let's look at the islets of Langerhans in humans and islets of Langerhans in rats and mouse. So the most fundamental difference is that the islets of Langerhans in rats have very high proportion of beta cells. The islets of Langerhans in humans have a very low proportion of beta cells. So this is one of the fundamental difference. The number of beta cells vary hugely. So in a rat model, the islets of Langerhans will contain 80% of the beta cells. In a human pancreas, the islets of Langerhans will only contain 40 to 45, maximum 50% of the beta cells. So this simple information, not very difficult to understand, the number of cells, the concentration of the beta cells in the islets of Langerhans. What this information tells us is that there must be a different kind of physiology undergoing in humans as compared to the rats or mouse, because beta cells and alpha cells, they have a physiological interaction. So if there are more beta cells, so it generally means that the beta cells will be dominant on the alpha cells in rat and mouse. Whereas in humans, the beta cells are low in number, so they will not be very much dominant over the alpha cells. So let's look at the microscopic images, which are aminocytochemically tagged. So instead of humans, we will use the monkey. And we call them non-human primates, okay? And there is not much difference between a monkey and a human, physiologically speaking. So the visual is not very great, but I will explain this to you. So staining, and you can see that in the rats, the islets are more visible as compared to in the monkey. The second image is the islets of Langerhans. They are stained with an anti-insulin antibody. So these images are stained with an anti-insulin antibody. So if you look at these islets of Langerhans, they are all of brown color. So it means that 80% of the cells are stained with the anti-insulin antibody. If we look at the monkey islets of Langerhans, we see that the central core there is no staining in the center, and there is a strong staining at the periphery. So this tells us that the beta cells are not present in the central area of the islets of Langerhans, they are present at the peripheral area of the islets of Langerhans. The next images are for the glucagon. So in these sections, we stain with the anti-glucagon antibody. And if we look at the sections from the rat, they give us the same picture as the picture of insulin in monkeys. The cells are present at the periphery. So these images are exact copies of this kind of image. So, and this image is an exact copy of this image. Majority of the cells have staying positive for glucagon in humans or monkeys. So there is a kind of a completely opposite situation going on in rats and in or again a different but much more clear. The first three images are, the first panel is of the hematoxylin eosin staining, the second one is the insulin, the third one is the glucagon, and the fourth one is the somatostatin. And you can clearly see that in the rats, the insulin staining is very predominant in the islets of Langerhans. The glucagon cells are present at the periphery. In the monkey, the beta cells, which are positive for insulin, they are actually present on the periphery. And the central core of the islets of Langerhans, they are made up by the alpha cells, which are glucagon positive. Okay, so this is a cytoarchitectural difference between rodent islets and human islets, or primate islets. This is also the reason why normally in animals diabetes do not happen. Diabetes is not a disease of the animals. If any person who is studying veterinary medicine, they never study diabetes, because diabetes do not happen in any animal. So diabetes is a specifically human disease. And this could be the reason for that naturally speaking, physiologically speaking, human islets, they do not have concentrated amount of beta cells as compared to alpha cells. Again, a different image, but same kind of staining. So over here, we also have a human section. So the first image, the A, is a human islets of Langerhans stained with anti-insulin, anti-glucagon, and anti-somatostatin antibody. Red is the insulin, green is for the glucagon, and the blue is for the somatostatin. So if you look at the first section, which is the A, which is from the human, almost all of the area is stained predominantly green, which is an indication that these are all alpha cells. Same situation is evident in the image B, which is from the monkey. So majority of the area is colored green because there are more alpha cells. But if we look at the C, which is the rat, the central area is hugely colored red because of too many beta cells which are present in the rat islets of Langerhans. Okay? So this is the cytoarchitectural difference. What could be the physiological difference? What could be the difference in the cell signaling and cell biology? There is a phenomena known as GSIS, and this phenomena is explained like GSIS goes for glucose stimulated insulin release. And this means that whenever we eat something, our glucose level will increase in our blood. This increase in the glucose level will force the beta cells to release insulin. So this phenomena is known as GSIS. So in the primates, this GSIS response is lower as compared to rodents. And the humans and monkeys, they have a higher basal insulin levels. The basal insulin levels means the normal insulin levels. When we are not eating anything, what is the normal level? So in rat and mouse, the normal insulin level is very low. As soon as these rat and mouse eat something, the insulin levels shoots up. This is not the case in humans. In humans, the normal insulin level is also not very low, and whenever we eat something, there is a slight increase in the insulin, not a very huge increase like in rat and mouse. The second fundamental difference between rat, mouse, and humans and monkeys is that the insulin stimulated glucose metabolism. So that metabolism of glucose, which is activated, which is stimulated by insulin. In humans and in primates, this metabolism happens in the muscles. So our muscles are the main site for the metabolism of the glucose. In a complete contrast, in rat and mouse, this main metabolic, this main metabolism of the glucose, this will not going to happen in the muscles. This will going to happen in the liver. So this is a huge difference. So 70 to 90% of this metabolism will happen in the skeletal muscles in the humans, and almost 90% of the glucose will be metabolized in the liver in the rat and mouse. So this is a huge difference. Third very important difference. Third very important difference. The ability of beta cells to proliferate is much more different as in rodents or in humans. Anyhow, the loudspeaker has stopped working. Anyhow, so the ability of the beta cells to proliferate, to multiply, is hugely different. It is hugely different in humans as compared to the rodents. In vitro proliferation of beta cells is only possible in rat and mouse. In vitro proliferation of human beta cells or monkey beta cells is nearly impossible. So this means that when we destroy beta cells in the rat and mouse, these cells have a natural ability to proliferate. So it is quite possible that after the first destruction, experimental destruction, these beta cells, they will proliferate. But this is not the case in humans or in monkeys, because the natural ability of beta cells to proliferate is very limited, and it is also time dependent. So during the early phase of development of the life, these beta cells will develop, but once the person has achieved the adulthood, these beta cells, they will fail to multiply. So this has a very significant impact on the medication, on the development of the medication for the diabetes. So this is another image. This is these are the isolates of Langerhans from a normal human being, type 1 diabetic human being, type 2 diabetic human being. Okay. So, and I will show you the specific differences which we need to look at. So this is a normal hematoxylin eosin stain section of the pancreas. This is showing the isolates of Langerhans in a normal human being. It is not diabetic, it's a normal human being. The second portion, the second image is type 1 diabetic human patient, and this is the isolates. Again, the blue color is for the glucagon. The light red color is for the insulin, and this gray color or light brown color is for the CD3, which is the we just compare, there is no staining for CBD. So there is no cytotoxic in type 2 diabetes. But there is a huge amount of amyloid. There is a huge amount of amyloid deposition, and this amyloid deposition indicates the dead beta cells. So there is a huge amount of dead beta cells and their cellular fragments which are still present over here. And when we stain for the beta cell, we see a huge area which do not have any cellular identity. So these cells are not positive for alpha cells. This area is not positive for alpha cells, not for beta cells. So only this is the alpha cell and beta cell positive area. So in a type 2 diabetic patient, almost 80% of alpha cells and beta cells they are gone. They are lost. And there is no cytotoxic. So this is one of the major differences between type 1 diabetic isolates and type 2 diabetic. So the question which comes to the mind that if we keep on working on the rodent models of diabetes, where we will stand in the next 10, 20 years? Because we have been working on these rodent models for the last 20, 30 years. And there is a clear, huge difference between the isolates of Langerhans, the main area which we generally focus in our diabetic research. And we use different types of herbal extracts, plant extracts, and we try to prove their ability to maintain glucose homeostasis, or we show their ability to, they help in the proliferation of the beta cells, which is good for rat and mouse. But rat and mouse, they do not have a problem of diabetes. Humans have, and humans have a very different isolates of Langerhans structure as compared to the rat and mouse. So it is just a food of thought, okay? And that will be the end. And thank you very much. And if you have any question, I hope you do. I will be happy to answer your questions. Lights on. हाँ जी, हाँ जी. इसका पॉजिटिव अगर हमें ये वजह से पॉजिटिव तो हमारे टाइपोडायबिटीज को सॉल्व करना. Some say it is a genetic problem because of some mutation in the genes of these immune cells, and they somehow start to attack specifically the isolates of Langerhans. How they are activated, we have no idea. How they are on a cell biological level, what is their main trigger, we have no idea. हाँ, अगर हमें आइडिया हो जाए तो we will actually solve this type of diabetic problem. Diabetes can cause obesity. Obesity can cause diabetes. They both are interchangeable. Glucolipotoxicity. When we have a high carbohydrate diet, so we have high level of glucose, and with that high carbohydrate diet, we are also taking high fat diet. So we have high lipids. So, and beta cells are specifically prone to high level of glucose and high level of lipids. Or this glucolipotoxicity actually ends up destroying beta cells. So both of them are interchangeable. Obesity can cause type 2 diabetes, and type 2 diabetes generally certainly can cause obesity. What is diabetes? अगर मैं आपसे पूछूं, what is diabetes? Diabetes is hyperglycemia. Hyperglycemia is what? High glucose. High glucose. Why high glucose? Because there is a lack of insulin. Okay. So if you can use your muscles to consume insulin, you can control it. If you restrict your carb intake, you can control it. Management, yes. Until unless we have the data, we do the experiment on primates and we see that. हाँ जी. On rodent models, because they're easy to have them, it's very easy to use them. In a very small space, you can have 50-60 rats or mouse. It is very cost effective. ठीक है. और